Hello everyone, I am Thuy. I come from Faculty of Food Science and Technology at Vietnam National University of Agriculture. Through this poster, I would like to introduce one of our research with the title Optimization of Pectin Extraction from Purple Passion Fruit Peel and Development of Pectin-Alginate Biofilm for Storage. As you know, a passion fruit peel accounts for a very high percentage of the total fruit weight, about 51%. This is an abundant source of byproducts, so taking advantage of this byproduct source to extract pectin will not only create valuable products for application in the food industry, but also contribute to solving environmental problems. So this study aimed to find out the optimal conditions for extracting pectin from purple passion fruit peel and to develop pectin-alginate composite biofilm by mixing extracted pectin in combination with alginate, glycerol, and calcium. To carry out this study, we used the following research methods. First, pectin was extracted using modified versions of the methods presented by Ragana et al. Second, central composite designs of response surface methodology was used to investigate the effects of three independent process variables, namely citric acid concentration, extraction temperature, and extraction time on the response is the pectin yields. These are the three factors that have the greatest influence on the effectiveness of pectin extraction. Each factor consists of 5 different levels, as can be seen in table 1. This is table 1, you can see here. And this is the form of the second order model. The second order polynomial equation explaining the relationships between the response and variables. Next is the method for analyzing the physiochemical parameter of the extracted pectin, degree, esterification, and pectin. A pectin sample was determined as reported by Project et al. Equivalent weight, methicillin index, total anhydrous acid content. A pectin sample was determined as reported by Jada et al. The next step after creating a biofilm from pectin and alginate, the specification of this biofilm was also determined, including tensile strength, elongation at break, water vapor permeability, and moisture absorption. After conducting the experiment, we obtained some results as follows. First, the results of 20 experiments are shown in table 2. This is table 2, you can see here. Table 2 shows the pectin yields extracted from passion fruit peel range from 13.74 to 23.91%. Furthermore, using the methods of least squares, the suitable quadratic model was found for the extraction of pectin from purple passion fruit peel. As you can see here, is the quadratic model. And analysis of variance indicates the model is significant. This regression equations so that three experimental factors and the interaction between the two factors have a significant effect on pectin yield, in which the temperature factor has the proportional influence, and it has the strongest influence. To optimize, it's necessary to first consider the interaction between each pair of factor on pectin yield. Response surface from figure 1 showing effects of independent variables on the yield pectin from passion fruit peel. Figure 1 here. Now, after that, the results are found the optimal condition to extract pectin from passion fruit peel as follows: citric acid concentration 8%, temperature 82.14 degrees Celsius, extraction time 81.07 minutes, and pectin yield reached 24.42%. Next, table 3 shows physiochemical characterization of pectin from purple passion fruit peel. This is table 3. You can see here. It has a different physiochemical properties compared to the market pectin. It also has shown that the extracted pectin is a low methylene pectin and is suitable for the purpose of biofilm formation for preservation. So in the next step, we made the Pectin-Alginate biofilm for the purpose of preserving fresh fruit. The results have determined the technical characteristics of the Pectin-Alginate biofilm in table 4, as you can see here. The technical characteristics of Pectin-Alginate biofilm indicated is the potential applications in the preservation of fresh fruits and vegetables. In summary, this study has 3 conclusions as follows. First, the generous quadratic model adequately explained the rate of variation and significantly represented the actual relationships between the independent variables and the response. Second, the optimal condition to extract pectin from passion fruit peel are as follows: citric acid concentration 8%, temperature 82.14 degrees Celsius, and extraction time 81.07 minutes for the highest yield pectin. And thirdly, these results open up a high potential application of Pectin-Alginate biofilm in the safe preservation of fresh fruits and vegetables. Thanks so much for listening. Thank you.